A furin cleavage site is the part of the spike protein that attaches to the cell and helps the virus infect cells. And it's very important for the kind of disease presentation you see, meaning the more virulent, the more severe disease that is caused is attributable to this furin cleavage site. Now, the interesting thing about this furin cleavage site is that it has not been identified in any other similar coronaviruses that infect humans. So many people thought that because it is such a rare finding, such a unique finding, including the sequence, the amino acid sequence, the codon sequence is so unique that many people thought that it had been engineered in a laboratory. So a furin cleavage site is an amino acid sequence that tells a certain enzyme to cleave the spike protein so that it changes its conformation and enables it to bind better to human cells. So let's say this is the spike protein. There is an amino acid sequence, 4 amino acids, that signal to this enzyme that it should cleave this site and open up in order to bind a human cell. So one arm is going to bind to what is called the receptor. There's an ACE2 receptor. And then the other arm is going to enable the virus to enter into the human cell. So that presence of that furin cleavage site not only helps it to become more transmissible, to spread more between people, it also makes the disease more severe. And because it was absent in other coronaviruses and similar viruses, it is a rare finding. So people thought it's been genetically engineered in the lab. However, most people argue and say that there is no proof that it was engineered. But even that is contested. You can make those mutations and leave no evidence. And so at this point, the ball can go either way. There is no clear case that the furin cleavage site is engineered or that it happened naturally. Something that would help us, it would be to find its presence in an animal host, and that has not yet been identified.